Bismillahirrahmanirrahim. Today I would like to share the knowledge about Cryptosporidium. My name is Sarfraz Rahman. I am a PhD scholar in the Department of Parasitology. I would like to share this knowledge with the references these books, Veterinary Parasitology, 2nd edition by G.M. Arkuhat, Veterinary Parasitology, 4th edition by M.A. Taylor, George's Parasitology for Veterinarians, 10th edition by D. White D. Bowman, a Handbook of Veterinary Parasitology, Domestic Animals of North America by Henry J. Griffiths, and one is the research paper that the name is Cryptosporidiosis, an overview by Gordon J. Licht, and next one is the Veterinary Parasitology Reference Manual, 5th edition by William J. Poirier, and last one is the internet sources. Please subscribe my channel and also push the button of bell icon. So the subphylum Sporozoa or Apicomplexa. This Sporozoa or Apicomplexa, this one is the subphylum, and it has the three main classes. The first one is Coccidia, or another name is elementary Sporozoa, and second one is Pyroplasmidia, and third one is the Hemosporidia, or another name is the blood Sporozoa. So the first one, that the Coccidia, it has the two main families. Number one is Hemiridae, and second one is the Sarcocystidae. So this family Hemiridae, it has the three main genera. The first one is Emeria, second one is Isospora, and third one is Cryptosporidium. While the Sarcocystidae family, that has the following genera. First one is the Toxoplasma, the second one is Neospora, and third one is Hemondia, and fourth one is Basnoita. So this class Pyroplasmidia, it has the following genera. That first one is the Theileria, Babesia, Cytoaxazone, and while the last class, that the Hemosporidia, it has the following genera. That first one is that the Hemoproteus, the second one is the Plasmodium, and last one is the Leucocytozone. Today I would like to share the knowledge about this genera, that is the Cryptosporidium. So now the Sporozoa or Apicomplexa. What means the Sporozoa or Apicomplexa? This one is the group has possession of an apical complex. So what is the apical complex? It is the structure which apparently assists penetration of the host cell, and it is visible only with the electron microscope, and having apical stages in their life cycle. So the trophozoites have no cilia and no flagella, and intracellular, these are the intracellular parasites, and it has the sexual stages. The name is that the gametogony, while the stage at which the gametes produce spore is called the sporogony, while the asexual stages, that is the name is schizogony, and this life cycle, these Apicomplexa divided into the three main classes. The first one is Coccidia or elementary Sporozoa, and second one is Pyroplasmidia, and third one is Hemosporidia or blood Sporozoa. So this first class, that the Hemiridia, that these are the intracellular parasites, and schizogony and gametogony take place into the same host. Sporogony or maturation take place outside of the host, and it has the three main genera. Their first one is Emeria, second one is Isospora, and third one is the Cryptosporidium. So I would like to tell and I would like to share the knowledge about detail of this Cryptosporidium. So what is the coccidiosis? And it is the infection of Emeria or Isospora, or collectively of Emeria and Isospora infection is also called the coccidiosis. And that one is the important point. Coccidia, these five coccidia have the different, these are different to each other. The first one is Emeria. It has the four sporocysts and two sporozoites, while the Isospora, that is the two sporocysts and four sporozoites, while the Cryptosporidium, it has the two sporocysts and four sporozoites. While the Toxoplasma, it has also the two sporocysts and four sporozoites, while the Sarcocystus, it has only one sporocyst and four sporozoites. So this one is the Cryptosporidium, and this parasite causes the disease. That name is the Cryptosporidiosis, or another name is watery diarrhea, or another famous name is the traveling diarrhea disease. So it is a zoonotic disease. And first description, this the parasite, the Cryptosporidium murus, that the name of the scientist, that is the American parasitologist. He described about this parasite, the Cryptosporidium murus, in 1910. While short name of this parasite, that the short name is of this parasite is the crypto, and host is the humans, mammals, birds, reptiles, amphibians, and fish. While the site of infection, the intestinal epithelium of the small intestine, and species Cryptosporidium have a lot of species, but it has divided into the isolates, and these Cryptosporidium isolates are assigned by the 19 important species. The first one is the Cryptosporidium parvum. It is present in the humans and mammals, while the Cryptosporidium humanus, it is predominantly present in the humans, while the Cryptosporidium murus, it is present in the rodents, farm animals, and humans. Cryptosporidium andersoni, that is present in the livestock and the humans, and while the Cryptosporidium belli, that is present in the poultry, and Cryptosporidium suis, that is present in the livestock, humans, and that the Cryptosporidium horari, that is present in guinea pigs and humans, Cryptosporidium phallus, that is present in cats and humans, while Cryptosporidium canis, that is present in dogs and humans, and last one is the Cryptosporidium meleagritis, that is present in the birds and humans. So the oocyst, size of the oocyst, especially the Cryptosporidium parvum, that is 4 to 4.5 micrometer, having the two sporocysts and four sporozoites, and oocysts are the two main types. That one is, first one is the thin-walled oocyst, that is excreted, thick-walled, that is excreted in the feces, while the thin-walled oocyst, that is released the sporozoites in the intestine, and then it creates the auto-infection due to the higher immunity. And next one is So this one is the life cycle of the Cryptosporidium, and Cryptosporidium have the complex life cycle, and its incubation period or prepatent period is 2 to 10 days, but average is the 7 days. And humans or other animals, first of all, that the two main oocysts, that the thick walled oocyst and thin walled oocyst. This thin walled oocyst, when enters or ingested by the humans or other animals, it is that the destroyed due to the immunity, and while the thick walled oocyst, that is the resistant. And this thick walled oocyst that produces the sporozoites, and these sporozoites changes into the trophozoites, and then trophozoites changes into the merogony, and that the type first meroant, this type first meroant then again changes into the merozoites, and these merozoites changes into the trophozoites, and these stages take place many times. And then again type second meroant, and then merozoites, then these merozoites changes into the two main gamete, macrogametes and microgametes, and these two gametes combine to form the zygote, and then zygote that changes into this sporogony, and this sporogony that ultimately produces the two main, that the thin walled oocyst or the thick walled oocyst. So this is the life cycle of the Cryptosporidium. What is the infective stage of this life cycle of Cryptosporidium? That is the oocyst containing the four sporozoites. One thing is more that is very important related to the life cycle, that is the humans or other animals, that you know that it is the zoonotic disease. So due to this humans, the infection spread, and that it take place the anthroponosis and zoonosis, or even in the environment the feces spread, and that can cause the infection to other humans or animals. So Cryptosporidiosis in humans and other different animals, the Cryptosporidiosis in humans, dogs, cats, birds, reptiles, amphibians, mammals. Cryptosporidiosis in humans. The most important species related to this Cryptosporidiosis in humans, that first one is the Cryptosporidium that huminus, Cryptosporidium parvum, Cryptosporidium murus, Cryptosporidium andersoni, and Cryptosporidium suis. That the host is the human beings, habitat inside the human being in form of the trophozoites, resides in the epithelial cells of the small intestine. Identification: the oocyst have the two main types, thin-walled oocyst or then the thick-walled oocyst. The thin-walled oocyst that is released in the intestine and causes the infection, while the thick-walled that is excreted in the feces. The parasite accidentally are swallowed that by the contaminated water, food, hands, and other stools. And the people affected, that the children, immunosuppressed people, travelers, and swimmers. So Cryptosporidium parvum, that this one is the Apicomplexa, and it is a coccidia, and the coccidia parasites are identified based on the structure of the oocyst. It is an obligate intracellular parasite, primarily infect the intestine. It forms the two oocysts that are the resistant to disinfections and even killed by the ozone and desiccation or 5% ammonium solution. You can see this in the slide, that the purple red color, that is this is for stain color, and you can observe this that the Cryptosporidium parvum. So the history in 1912 that the discovered this Cryptosporidium parvum by the Ernst Edward Teiser, and this one he was an American parasitologist, and the outbreak associated with this, the drinking water, food, swimming pools and lakes, hospitals, nosocomial infections can be take place, HIV wards and pediatric hospitals. These are the source of spreading this infection. So the 1993, the milk whale World War WI, that the largest known water supply outbreak, and 40,000 persons become ill. And while in 1997, the Minnesota Zoo decorated a water fountain that infected different animals, and 369 cases, most cases in the children that the less than 10 years old, that are infected with this decorated water fountain. Morbidity or mortality: North America, the 2% of the population infected and 80% previously exposed. In the worldwide, the prevalence lower in the developed countries, 1 to 1.5%, and higher in the developing countries, 3 to 20%. And animals morbidity high while the mortality is low. Transmission: fecal oral route, the person to person, person to animals, waterborne, foodborne, mechanical, and aerosol. It is a rare. The clinical signs: the symptom of crypto that generally begins 2 to 10 days, average 7 days after becoming infected with this parasite. Symptoms including first one is the watery diarrhea, that is very important and most critical sign. The stomach cramps or pain, dehydration, nausea, vomiting, fever, weight loss. Symptoms usually last about 1 to 2 weeks, with the range of the few days to 4 or more weeks in the people with a healthy immune system. Most common symptom of the Cryptosporidiosis is the watery diarrhea. Some people with crypto will have no symptom at all. So the diagnosis: lab diagnosis, direct microscopy, or Nielsen technique or Aurea technique, and you can observe the sporozoites granules, bright red, and biopsy. While related to diagnosis mode, that the is for staining, autofluorescence, biopsy of intestinal mucosa, and PCR. And the treatment: that the first one is the spiromycin, 500 milligram per every 8 hours, halofuginone, 60 to 125 milligram per kg body weight daily for 7 days in dogs and cats. The parvum, 150 milligram per kg body weight single and dose for 5 days, and azithromycin, that 5 to 10 milligram per kg twice a day for 5 to 7 days for dog. And the control and prevention in humans: wash the hands properly, wash the fruits and vegetables, don't swallow the water from lakes, streams, public pools, or minimize the contact with the young animals. Cryptosporidiosis in animals: all mammals, common in calves and lambs, other species, pigs, dogs, cats, horse, these are the rares. And signs and symptoms in animals: incubation period almost 4 days in calves, anorexia, diarrhea, tenesmus in the feces, and weight loss. And postmortem lesions in animals: the gross lesions, hyperemia of the intestinal mucosa, and yellowish intestinal contents. This one is the yellowish. And microscopic lesions: mild to severe villous atrophy and spherical organism in the brush border. And the diagnosis in animals: differentiation: you can differentiate from the Giardia, Isospora, Microsporidia, Salmonella, Rotavirus. And the diagnostic test, the fecal flotation, acid fast staining, immunofluorescence, these are the tests. The treatment in animals, the no specific treatment, it's just only the supportive care, and there's no proper vaccine. Okay, that the control and prevention in animals, keep the sick away from the healthy animals, clean and disinfecting the pots, and give the good nutrition. It is also helpful in the prevention of this disease. Okay, thank you so much.